Cancer is this huge problem, and so what if I started looking into ways of solving this problem? Virus-like particles could be cheaper. We're not really at a point where virus-like particles are nearly as good at entering cells and delivering DNA or RNA as viruses are. Putting a AAV inside of a protein vault is that it shields it from the pre-existing antibodies that humans produce. People have tried immunosuppression of everything, but it can be more dangerous and more problematic. Specifically suppressing cellular immunity, it's a lot more straightforward of a procedure and a lot safer.
Body. What is it?
Fleming of St Mary's Hospital, London, who discovered that it produces the drug known as penicillin.
The question was, which drug would go with which cell?
It's a collaboration that's unexpected because it's between humans and the tiniest organisms.
The howl of a dire wolf.
Dorset lamb may represent a major landmark.
I did not start off planning to design proteins.
Scientific breakthrough will probably be something that we hard to imagine.
Test at birth could map out an individual's entire future risk of disease.
Take us through the through line, right, of your scientific journey. How did it kind of start? How did you become interested in science in the first place?
Yeah, so basically as long as I can remember, I've been interested in science. I think when I was, in, preschool or something, I actually built a submarine, not an actual submarine, but like a sort of a structure that I, did play fantasy submarine adventures in. And ever since I've been super young, I've always just been fascinated by science. When I was in elementary school, I wanted to be a theoretical physicist. But once I was in middle school, I discovered the world of biology and biotechnology, and I found I had this super strong affinity for it. And I mean, even before that, also as long as I can remember, basically, I've had this very strong conviction that I'm here to make a difference and I have something to do here on this earth. And so I guess what happened was that it sort of evolved into me realizing the power of biotechnology and what can be done with biotechnology. And I also have this very strong creative streak. And so when I discovered synthetic biology, I actually, even before that, had sort of the idea of, well, what if you did this type of thing with biology? And then I heard the term synthetic biology, probably on Wikipedia or wherever, and I was like, this is what I'm doing. This is what I want to do. And so I was in middle school, like 7th or 8th grade, and I was like, okay, well, cancer is this huge problem. And so what if I started looking into ways of solving this problem? And so I started to generate hypotheses of, what's a design that I could use to treat a wide array of cancers. And I actually, when I was in 8th grade, I did this sort of independent pseudo-research project where I just came up with a bunch of technical ideas or semi-technical ideas that were aimed at treating or curing cancers. And from there, I ended up transitioning into antimicrobials. And that's sort of where my real scientific journey begins. That's where I started doing more formal research. When I was in 8th grade, I came up with this idea: what if you could fight antibiotic resistance by causing chaos in bacteria, by creating these aggregates with a bunch of proteins stuck together inside of bacteria that would just disrupt their systems and cause them to cease functioning? Because a lot of antibiotics, what they do is they bind to a very specific pocket on functional structures within bacteria, like ribosomes or polymerases, and they block those structures from working. But even small changes in mutations that can happen in these structures, even small changes can then reverse the ability of the antibiotic from actually working. And then you've got antibiotic resistance. And so I thought, okay, well, what if you could just affect everything at once? They wouldn't be able to mutate away from that. And that's how I got started with my science fair project, which was my first real research project that I was working on in a lab. And I started in a lab in 10th grade. And that's sort of where my scientific journey began.
Yeah, very, very precocious, starting a submarine in did you say elementary school?
I think that was preschool. It was, like I said, it was really just a a structure where I think I used a bunch of blocks to build up sort of a cave-looking thing, and then play acted as if it was a submarine going on adventures. But it was fun. It was like I said, I've always had a creative streak.
Talk to us a little bit about sort of creating antimicrobial peptides. This seems to be one of your first big breakthroughs. How did the project manifest?
Yeah, so as I said, I initially came up with the sort of base idea in 8th grade. I wanted to. I actually had been taking some classes at the high school that I eventually went to, and I saw that there were a lot of young scientists there doing real research, doing science fair projects that were actually working in labs. They were doing graduate level research, and one of them was doing research on antimicrobials. I think it was a nanoparticle-based antimicrobial project. And I thought, okay, so I'd been working on, thinking about cancer, but here's this other problem that's a really big problem, and I think that could have a lot of impact. And but it also is something that I feel like the resources that I can acquire at this time in my life could lead into very reasonably. And so I ended up going in that direction, and I really loved it because I got to work with synthetic biology. I got to implement this idea of causing chaos in bacteria using an aggregating antimicrobial peptide. And that's sort of where the idea of this antimicrobial peptide evolved from, something that can glom together and form these big aggregates in bacteria and kill them. And from there, I started doing my science fair project, which was based on that. So I developed the peptide itself, which was based on the principle of hydrophobicity primarily, as well as a few other ideas. So essentially this peptide started out as something that was highly hydrophobic. So when it's in water, the thermodynamics of this lead to the hydrophobic regions sort of wanting to get away from the water. And so to do this, they pack themselves together and bury themselves beneath the more hydrophilic parts of the peptide. And so if you produce a lot of this peptide inside of a bacterium, it produces these aggregates, which I actually later was able to visualize with fluorescence microscopy. And the hydrophobicity was sort of the main principle by which it aggregated. I actually had a control peptide that had a lot of the hydrophobic regions interrupted by charged regions, and that didn't work nearly as well. But I also made several design tweaks. So I excluded certain amino acids that were known to be bound by molecular chaperones, which are molecules, which are proteins in bacteria that help the folding of proteins. And the idea here is that we want the proteins to misfold. We want them to cause chaos. And so to avoid the molecular chaperones from reversing the process, I specifically excluded certain motifs or certain sequences from the peptides, even certain amino acids. And that was sort of the design principle behind it. And from there, I went on to design a delivery system, because in order to make this peptide inside of a bacterium, you need to get DNA that encodes it into the bacterium. And what I did was I used a process that bacteria naturally do called bacterial conjugation, where one bacterium carries a circular piece of DNA called a plasmid and transfers that to another bacterium using an array of proteins that are encoded by that plasmid. And so essentially DNA moving between bacteria. And bacteria actually normally use this to produce, to transfer useful genes that are useful to their own fitness, like antibiotic resistance genes, ironically enough. But I was using it sort of to attack the bacteria. So I developed a way of making my gene, my antimicrobial gene, which encodes the peptide OPAL, overexpressed protein aggregator lipophilic. OPAL was expressed by this gene, so it's the OPAL gene and the OPAL peptide. Mm-hmm. And this gene was dormant in the donor bacteria because I used a switch, a promoter, which is only active in the target bacteria. So it can only turn on in the target bacteria. And then I was able to use that to sort of mix donor bacteria carrying this toxic gene with target bacteria, and I showed that you can transfer it via bacterial conjugation, via this movement of DNA from one bacterium to another, whereupon it turns on, produces the aggregating antimicrobial peptide, and kills the target bacteria.
Fascinating. And how does a switch work?
So a promoter is it's basically the sequence of DNA that proteins in a cell bind to turn on a gene. So it's a sequence that several different proteins will typically bind to. And in bacteria, the specific promoter I use is called T7. It is a very strong promoter, so it produces a lot of protein, which was useful for my goals. And it's only active in a specific strain of E. coli. That encode a protein called T7 RNA polymerase, because T7 RNA polymerase is needed to turn on to produce the RNAs, which produce the proteins in the bacteria that carry this gene. So that's essentially how it works. But there's a lot of promoters. Promoters are everywhere. We have tons and tons of promoters for our own proteins. Bacteria use lots of different promoters for their proteins. Even viruses use different promoters. And so using a promoter to target something is essentially taking advantage of the machinery that the cell has to turn on the promoter.
Fascinating. And if you had to apply this in a real-world context, obviously we have a large number of increasingly antibiotic-resistant bacteria in our day-to-day world. How do you actually package this technology and deliver it to a real-world bacterium that's resistant?
Right. So it kind of depends on the location of the infection, because if it's in the blood, you're probably not going to even want to use a bacterial conjugation strategy at all. If it's a bacteremia in the blood, because that's just introducing more bacteria into the blood. Even benign bacteria aren't supposed to be there and can cause problems. However, if it's an infection that is in the gut or it's a wound infection, that would be a prime area to use bacterial conjugation, where you would introduce donor bacteria to the gut or to the wound, and they would spread this gene not only to the target pathogen, but also to your indigenous microbiota, the indigenous bacteria that live inside you. And the indigenous microbiota, you could design the promoter so that it doesn't affect those bacteria, so that your own bacteria aren't harmed by it. It doesn't turn on the toxic antimicrobial peptide gene. And so then the indigenous microbiota themselves, they become the donors. They become more donors. And so my vision for this was essentially that you could really increase the frequency with which the antimicrobial gene is transferred into the target bacteria in that way. And then one more thing is that if you are facing a bacterial infection that's somewhere where you can't introduce donors, I suppose you probably could in a lung infection, depending on the context, but a blood infection or something like that, I would actually use bacteriophages to deliver the gene. Phage therapy is an emerging field, and it has a lot of challenges to overcome still, but it's advancing very rapidly. And hypothetically, you could deliver this antimicrobial peptide gene in a bacteriophage, which is a virus that infects bacteria, for those of you that aren't aware. And then the bacteriophage would deliver the gene to the cytoplasm of the bacteria.
Fascinating. Okay, so you engineer this donor bacteria by inserting a plasmid that encodes this antimicrobial peptide that is very hydrophobic, that sort of causes chaos in any bacteria that it's within, but it's entrusted or sort of encoded beneath a promoter or sort of downstream of a promoter sequence, which only produces this antimicrobial peptide in specific bacteria based on your promoter design, right? So then you introduce this donor bacteria into your throat. For example, if you have a throat infection, and this goes and through conjugation, through sort of basically promiscuous sharing of its plasmid DNA with other bacteria, introduces that antimicrobial peptide, but now it's under a different promoter. Can you sort of explain how that works?
Yeah, so the promoter is actually the same within, it stays within a plasmid that encodes it. So this plasmid has a specific promoter, and that specific promoter, the whole plasmid is transferred into the target bacteria. However, this promoter, the machinery of the donor bacteria, and hopefully most of your indigenous bacteria, the machinery that they have, the proteins that they have, can't turn this promoter on. So you have to choose a promoter that's specific to the the target bacteria.
Okay.
One more thing I should mention is that the promoter I used is a very strong promoter that's typically used in laboratory contexts. And so it wouldn't necessarily be the promoter of choice in a real-world context. However, what one could do is one could encode the T7 RNA polymerase under the control of another promoter, which then produces T7 RNA polymerase, and maybe is a weaker promoter, but allows the stronger promoter to function in bacteria, because we want to produce as much of this antimicrobial peptide as possible.
Mm-hmm. Okay, fascinating. Great. The other thing that you're much more, I think, known for, or at least in the zeitgeist and in the sort of Foresight Fellowship groups, is delivery technology, right? You're the co-inventor, potential inventor of protein vaults for delivery of essentially anything. Talk to us a little bit about, what are protein vaults, and then we can go back and talk about the ancestry of the entire delivery space as well.
Sure. So I should say that I'm the inventor of specifically protein vaults for delivery of viruses. And just to sort of clarify that first, but as to what protein vaults are, so protein vaults are a mysterious organelle. They're composed of 78 copies of a protein called major vault protein. They look like two cups stuck together that have sort of a specific structure. And these cups are they're very nice. They have sort of a tapering structure on the ends. In a way, they almost look like little cathedrals or little mosques or something like that have been stuck together like a couple of domes. And they essentially have an interior compartment that's very large. But they're composed entirely of this protein, major vault protein. And the interesting thing is that most eukaryotes produce them in large quantities in their cells, including humans. And in humans, the protein vaults also package a few other proteins and a few small RNAs. In a delivery context, we actually get rid of those for the most part. But protein vaults are this really beautiful organelle which does not have a known function. People have tried a variety of things to try and figure out the function of vaults. They're known to be upregulated in certain cancers, which is kind of weird. They're also known to be like if you infect mice with a bacterium called Pseudomonas aeruginosa and you have these mice be deficient in the production of major vault protein, so they can't produce vaults, they actually die more frequently. So it may have some form of immunological role, but we don't really know. The nice thing about vaults, though, is that because they're produced in such large quantities within human cells, they are bioinvisible to the human immune system. They're recognized as self. And what really my big contribution was recognizing that vaults are not only useful as a delivery system for small things like peptides or small molecules, but it's possible to combine sort of the trails of thought in the gene therapy field and the protein vault field to realize that AAVs, or adeno-associated viruses, which are one of the most popular and one of the best delivery systems that has been developed to date for gene therapy, AAVs are, despite their advantages, between 30 and 60% of people have pre-existing antibodies which get rid of them. And because of this, if you've been exposed to AAVs at some point and you've produced antibodies against them, then you can't get the treatments that could potentially save your life. And so if you have a disease that's treatable by an AAV gene therapy, you actually have to undergo testing to see if you have these pre-existing antibodies, and if you have them, you're not eligible for the treatment. And so what I did was I made the connection that, hey, wait, vaults are big. They're big enough to fit an AAV inside of themselves, about 70 nanometers long and 35 nanometers wide, whereas an AAV itself is about 25 nanometers in diameter. And originally, I actually wanted to put multiple AAVs in to increase the amount of DNA that could be transferred to a single cell, which as it turns out, is possible, but happens very infrequently. At some point in the future, I may revisit that idea and try to increase the frequency with which it happens. But the main advantage of vaultAAVs, or putting a AAV inside of a protein vault, is that it shields it from the pre-existing antibodies that humans produce. Because remember, the vault itself is bioinvisible to the immune system, and so antibodies, they're not going to be able to penetrate it and reach the AAV. It essentially acts as a Trojan horse, where cells will take up the vault that contains the AAV, and then the vault falls apart in the structure called an endosome, which is essentially the bubble, the lipid bubble that cells use once they've eaten a particle. The endosome actually, cells tend to acidify the endosome via a variety of mechanisms involving something called VATPase, but that's a technical detail. And so when the endosome forms, it starts to acidify, and it eventually will become a lysosome, which destroys things. However, in the case of AAVs, once an AAV is in an endosome, it has a way of escaping the endosome. It actually breaks down the wall of the endosome and then goes straight to the nucleus to deliver the DNA that can be, in the case of a gene therapy, life-changing. And so the process is essentially you'll deliver these vaults that shield the AAVs from antibodies. The cells will eat them, and then you'll be able to deliver the DNA into the cell and into the nucleus, thus circumventing the pre-existing immunity that's found in so many patients.
Incredible. Let's take a step back to discuss the wider space of delivery and how it's evolved over time. This is a somewhat very novel contribution to the space. The space has been around for a while, right? We've had AAVs, we've had LNPs for quite some time. Only very recently have they entered the public zeitgeist through the mRNA vaccines, and then recently baby KJ. Talk to us about what was the first delivery mechanism that you were aware of, and what was how did the field sort of progress since then?
Yeah, so as to the first delivery system that I was aware of, I think I actually probably became aware of AAVs first, although I don't believe that was the first actual gene therapy delivery system that was used. In fact, adenovirus was used earlier than AAV. It was more popular around 25, 30 years ago. And so adenovirus is a different virus. It's a bit larger. It's about 100 nanometers in diameter, and it has these fibers which sort of point off of it that make it look like a Sputnik, kind of. But they're pointing away from the center. And adenovirus was originally the vector of choice, but a patient who received adenovirus early on in the days of the field, I forget the exact year, but it was around 2000. This patient, Jesse Gelsinger, he actually died, and it was a very mismanaged clinical trial. And he received too high of a dose of adenovirus, and he had a very strong immune reaction to it, and he died. And adenovirus has since been less popular. However, that's not to say that it's necessarily worse, because AAV itself has had a number of patient deaths in recent years because people have been using very high doses of AAV. And it also has its own drawbacks, like being smaller and not carrying as much DNA. But adenovirus was one of the more popular gene therapy delivery systems early on. And then, of course, LNPs became popular through the COVID vaccines. They had been used before, of course, but the COVID vaccines, I think, them being given to millions and millions of people really propelled their popularity. Then there's also lentivirus. So lentivirus is another virus that is primarily used for ex vivo treatments, which means that you take cells out of a patient and you treat them with the virus to deliver some DNA, and then you grow some of the cells that received that DNA and put them back into the patient. And that's actually used for a lot of cancer treatments for enhancing the immune cells, T cells, that are able to attack cancers. So these are the main delivery vehicles that are around. But since then, there's been some additional more exotic ones like proteolipid vehicles and so on and so forth. So yeah, there's definitely a panoply of delivery systems. And what I've noticed is that they all have benefits and drawbacks. And one of the interesting things about adenovirus that I'd like to mention is that it does it actually is possible to use at much lower doses than AAV because it enters cells much more strongly. It transduces them. That's what it's called. It's taken up into cells much more strongly. It also has sort of stronger gene expression once it's there. And so you can use lower doses of it, but it's more toxic overall. And since the early 2000s, there's been a lot of development in adenovirus to make it safer. And so I think adenovirus also has high potential. I've had some interest lately in potentially leveraging adenovirus for gene delivery for mental health treatments, because adenovirus was actually used in some of the sort of secondarily popular COVID vaccines by AstraZeneca, and it was administered to I think around 30 million people. And there were very rare side effects, and for the most part, it was safe. However, one of the big things about adenovirus is that it doesn't cross the blood-brain barrier. And so something that I'm interested in, and I haven't done yet, but I'm working on, or I'm working towards, is to develop a way of making adenovirus cross the blood-brain barrier to take advantage of its manufacturability, which, because of the way that adenovirus replicates in certain cell types that are, you can actually grow certain cells and make adenovirus in them. Because of its sort of continuous replication in those cells for a variety of reasons, the cells actually encode all of the what's called helper functions that are necessary to make the adenovirus replicate itself, whereas in AAVs, you can't do that. You have to deliver 3 different plasmids to the same cell to make an AAV, and then hope that the cell makes as much AAV as possible and then harvest immediately. Whereas with adenovirus, you can just keep growing over and over again, which is why the adenovirus is potentially more scalable than AAV for these sort of large patient populations. And so I have an ambition to engineer adenovirus to cross the blood-brain barrier as a way of potentially treating very widespread and common mental health indications like anxiety and depression.
Through genome engineering.
That's right.
Welcome to the Frameshifts podcast, where we spotlight the world's most ambitious biotech founders, investors, researchers, and changemakers. This episode is brought to you from the offices of Polsinelli, a national law firm that helps biotech companies do everything from incorporation to patents, trademarks, and venture financings. Thanks to Polsinelli for being part of the growing ecosystem of scientists, investors, and builders shaping the century of biology.
We touched a little bit about the pros and cons, sort of comparing and contrasting these tools. I find that so many people like to hate on AAVs, and that the entire gene delivery kind of ecosystem and community, it operates very much on vibes and not on actual science. Like whenever a patient dies from something, that's not hot at all anymore. You can't be doing AAVs, and no attention paid at all to like, what was the dose used? Like, right. Can it be used safely? It's sort of similar to the reaction to nuclear energy generation, right? It's like, there was Fukushima, and that's horrible, and we don't like nuclear weapons, so we can't do that, and then it's sort of like, well, but nuclear energy generation, or energy generation using nuclear power is actually like quite safe now, and if you look at it sort of logically and from first principles, it makes sense to keep doing it. I think a similar case can be made for AAVs, like. It doesn't matter that a couple patients die. That's tragic. But what matters is that, is science on your side? Is there a better delivery vector right now? And there doesn't really seem to be. Talk to me a bit about that. Like, am I correct? Because I'm not actually that familiar with the space. But is this sort of general vibe of like vibe shifting from one vector to another sort of accurate?
Yeah, I would definitely agree with that. It's definitely a matter of reputation of the vectors. And to be fair, I mean, there obviously, if there's a sort of a trend of several things happening in a row, I think that there's something to be said for the evidence as well. But I do think that like you said, the vibes play a bigger role than they really deserve to in this. And that's sort of because vibes really do matter in business, in entrepreneurship, because investors aren't necessarily going to be familiar with sort of all the technical details. Some investors do. Some investors bring on consultants who will sort of interview people ahead of time. But there's sort of this general this general lack of extreme specialization, which of course makes sense because not everyone can know as much as the scientists actually studying the vectors. And so people have to operate to some degree on the reputation of the vectors. And as unfortunate as it is, that does happen. And so it makes sense that this happens, but I do think that there needs to be more communication of sort of, like you said, the dose and the effects of how it was handled, what drugs were given to the patient, perhaps immunosuppressants of some kind, if those were used in an appropriate fashion to sort of combat the immune response that happens against the virus. I think this very much applies to both AAV and adenovirus because they've both faced criticism for their immunogenicity. Although AAV for many years had sort of been in the limelight with regards to having a reputation for a lower immunogenicity, it's now starting to catch up to itself, and we're seeing people criticizing the immunogenicity of AAVs. But the thing is that people are using very high doses, and the reason is, of course, that in order to achieve therapeutic efficacy for a lot of these like protein production treatments, so for like metabolic diseases and stuff.
Like when you're missing a particular protein.
Right. You need to have a lot of that gene in your body to actually replace it properly. However, that's not necessarily good. And so I think that I think that some of the things I've been working on could potentially help with this. For the AAVs, of course, there's the vaultAAVs, which they are because it shields from antibodies, the whole immune response should end up being dampened because antibodies are kind of linked to a chain reaction of events that can lead to higher immunogenicity. There is what's called cellular immunity, which is once the AAV is inside of cells, the immune system can recognize those cells that have been infected with the AAV and try to get rid of them. But there are very well-established methods for suppressing this using small molecule drugs that are pretty simple and safe. I think vaultAAVs have a lot of potential for this, which is why I actually spun off a company from my work on vaultAAVs. I co-founded it with my PI, David Curiel. And what we did was essentially it's called Cathedral, based on sort of the structure of vaults and how it looks. And this company, initially we'd been shopping it around to some investors, and we actually found another company that's currently in stealth. And that company, we merged with that company, and so now we have a lot of resources with which to really propel this technology forward and make it into something that's not just a proof of concept on the bench, but something that really will hopefully become a clinical reality eventually.
Mm-hmm. There are also alternative strategies for avoiding the immunogenicity problem with AAVs, right? Like especially antibody-mediated immunity. You can enzymatically digest the antibodies just prior to delivering the AAV intravenously. Right? What's sort of the pros and cons of doing that as opposed to delivering this via a vaultAAV?
Yeah, so when you're digesting large numbers of antibodies in the circulation, this can be a viable strategy, but it also can be potentially disruptive to the immune system in a longer-term fashion. We don't know exactly what's going to happen if you get rid of that many antibodies that quickly. And then that can also lead to its own immune reactions, of course, because antibodies themselves can be immunogenic and the enzymes can be immunogenic. However, I wouldn't say it's a bad strategy. I think it's actually quite a good strategy. Mm-hmm. The advantage of vaultAAVs is primarily that it really stealths the AAVs completely, so you don't have to mess with the body as much to deliver it. And in fact, there may be certain tissue types that actually take up vaultAAVs more efficiently. We don't know this yet because we're still in the process of doing some experiments, but there may be certain tissue types that take up vaultAAVs more efficiently than others. Mm-hmm. And in fact, in one of the early experiments I did in the lab, there was about 4 or 5 times as much transduction, as much uptake of the vaultAAVs into cells as it was for the formulation that lacked vaults, just the AAVs alone, which is interesting. And that was independent of the antibody shielding effect. So I saw both a shielding effect and an effect in this one cell line, CHOLEC2, which actually enhanced the uptake of the vaultAAVs relative to AAVs alone. And while this may or may not persist in the body, this is something that we're definitely investigating, and I think for certain tissues may still occur. And the reason that that's important is that if you're able to get the AAVs into the vaultAAVs into the tissue more efficiently, you can actually use a lower dose overall, which will increase the overall safety as well as decrease the cost. Because a big challenge with AAVs is manufacturing, and the cost of AAVs can be quite high. And so this would sort of be a win-win across the board.
Mm-hmm. Okay, interesting. I want to come back to the manufacturing point and sort of how to get this at scale, and also why we're doing this all in the first place, and what's the sort of transhumanist future that we want humanity to get to.
Yeah.
But let's touch a little bit more on the other strategies here, right? I mean, there are other bets being made on virus-like particles, like very, and also like very stripped down versions of AAVs that you may not even be able to still call an AAV, right? It's incredibly stripped down, maybe kind of similar to a VLP or a virus-like particle in that way. What are the other bets being made in the space, including these, and sort of touch on their strengths and disadvantages and where they're going? What's the hope for them?
So one of the biggest bets being made in the AAV space, or in the gene therapy space in general, really, is using a combination of directed evolution and machine learning to engineer AAV capsids to circumvent the immune response. And this is being pioneered by a company called Dino Therapeutics, which was spun out of George Church's lab. And I really admire what they're doing. I think it's very, very promising. However, the thing about this is that if you engineer the capsid by mutating certain combinations of amino acids and all of that, and you find which capsids are able to escape the antibodies through this mechanism so that the antibodies can't stick to the capsids, what happens is that you end up with a system that can be used once, but not multiple times. And even if it's used once, there still can be some potential for antibody interference because of cross-reactivity between different types of antigens. So I think it's a very promising strategy, but I think it still, to some degree, has yet to prove itself because it's kind of been being pushed in sort of the mid-stage startup kind of thing, where they have a lot of funding, they have some partnerships with companies, but to my knowledge, I don't believe they actually have any clinically approved AAV products that are directly based on their capsids. I think they may have some clinical trials going, though. And so there's some question about that. But sort of the main point I want to return to is the redosability point. And one thing that my company is sort of betting on is that we can redose vaultAAVs, so use it multiple times, which could be very beneficial for a variety of reasons. So it could actually decrease the necessary dose of vaultAAVs that's needed, so you don't need as many vaults and as many AAVs, which would decrease the cost and decrease the toxicity, as I mentioned earlier. And you could do this by spacing out doses over multiple doses by having a vaultAAV dose once, twice, maybe even 3 times, and sort of achieving the therapeutic effect via the cumulative effect of all of these doses. And so you can't do that with an engineered capsid because in order to do that, essentially you would need to have multiple engineered capsids that go through independent approval processes for every single dose you do. And I think that this is one of the big benefits of vaultAAVs.
So the core premise being that regardless of how much engineering you do to escape immunogenicity, these are fundamentally non-self epitopes that are encompassing this AAV capsid. And so given the sort of FDA's very rigorous approval process for every new kind of or every single engineered substance essentially needs a new round of approval, that you will not be able to redose this, that it escapes immunogenicity, but then it's recognized as a non-self epitope, and there will be a learned sort of response that the immune system will then generate for subsequent doses.
Exactly, and that's why I think that the redosability potential of vaultAAVs that we're hoping to start testing sometime in the next few months could really be a big game changer for the gene therapy field.
Mm-hmm. Now, the other thing I want to come back to is something you mentioned earlier around cellular immunity, right? Like these capsids still fundamentally get digested. The peptides get presented on MHC class 1 on the outside of the cell membranes, and then the immune system recognizes these again as non-self. And so there's redosability in terms of humoral immunity, but maybe not cellular immunity.
Right.
What are the strategies around overcoming the cellular immunity problem?
Yeah, so cellular immunity is definitely something that I have some concerns about. However, there are very well-established pharmaceuticals, steroid-based drugs actually, that can be used temporarily, that are reasonably safe and can suppress the cellular immune response in a transient fashion. And so what you could do is when you're getting a dose of AAV, you would first take this small molecule, probably a pill of some kind, or an injection, I suppose, and you would be able to have this sort of mild temporary suppression of one form of immunity. In the case of antibody-based suppression, it's a lot more difficult, and it can have more negative side effects. And so there's still been, people have tried immunosuppression of everything, but it can be more dangerous and more problematic. Whereas in the case of specifically suppressing cellular immunity, there's it's a lot more straightforward of a procedure and a lot safer.
Mm-hmm. Can you also touch on virus-like particles?
Yeah, so virus-like particles, I think, are potentially the longer-term future of gene therapy. So virus-like particles are really cool in that you can design these sort of protein cages to carry your DNA or RNA and have them deliver the payload rather than using a virus, which, of course, could potentially be more immunogenic because it's been sort of the immune system sort of primed to recognize viruses because we've been fighting viruses throughout our evolutionary history. Whereas virus-like particles can be designed, again, they can be designed in certain ways. I mean, if you just design a random virus-like particle, it probably would still be immunogenic, though perhaps less so than a virus. But you can design virus-like particles to be minimally immunogenic. And there's a lot of new tools based on AI that have come out recently, a lot of them from David Baker's lab at University of Washington, Seattle, or Washington University, Seattle, which can be used to design sort of de novo, completely new protein cages. They're virus-like particles. And so I think they have a lot of promise. I think that they could evade immunity. I think that they could be less expensive to make because you could potentially make all of the proteins needed in bacteria, in E. coli, which would be much cheaper than making viruses that require mammalian cells or potentially even insect cells. Actually, AAVs can be made in mammalian or insect cells, just as a clarifying point. But both of those can be expensive, and the entire process behind making AAVs is quite expensive for a lot of reasons, in part actually involving plasmid DNA, but I digress. The point that I want to make is that virus-like particles could be cheaper. The big drawback, though, right now, and I think but perhaps temporarily, is that we're not really at a point where virus-like particles are nearly as good at entering cells and delivering DNA or RNA as viruses are, because virus-like particles are sort of these clumsy, kind of human-designed systems that haven't really been fine-tuned over billions of years of evolution to enter cells. So we don't have yet on our side the mechanistic power that viruses bring from their evolutionary history. And so virus-like particles are a very cool idea. They're very interesting, and I think they have a lot of potential. But like lipid nanoparticles, actually, they just don't have the transduction power yet that I think is necessary to really propel them to be at the center of gene therapy.
It's fascinating. There essentially seems to be two strategies that are all converging on the holy grail of delivery. One is stripping down what nature has already provided us to escape immunogenicity, right? Which is the AAVs, the adenoviruses, etc. And then the other approach is assembling ourselves de novo using engineering these like minimally capable delivery vectors that and then trying to engineer them so that they have high transduction capability. And then there's this other approach where you're almost like taking advantage of this somewhat obscure existing protein vault that our bodies already produce. That it seems quite versatile. You can hide a lot of things in there. Yeah,
Yes.
Potentially, I'm not sure if it's large enough to fit an adenovirus, is it?
Unfortunately not. It could potentially be possible to coat adenoviruses with vaults, but that may interfere with the adenovirus's transduction abilities. I've had some ideas in my head around playing around with that, but I haven't had a chance to explore them.
Got it. So I think we're converging a little bit. That there's synthetic biology as a discipline. There's like the delivery vectors. There's the gene editing tools. There's gene therapy as a collective. What are all of these exponentially accelerating technologies converging towards? What's the future that we're hoping to enable?
I mean, I think that, like I said about virus-like particles, I think that having some convergence of the ability to design biology in a way that allows for dynamic motions that are highly sophisticated and can sort of replicate the abilities of viruses while being de novo and designed to circumvent immune responses at the same time. I think that would really be sort of the ultimate gene delivery vehicle. If you could design a virus-like particle that's easy to produce, that has that circumvents immunity, and that has the sophistication in terms of mechanistic capacities that it can do what viruses do, and enter cells and escape endosomes and traffic to the nucleus and express and allow the DNA or RNA to express proteins in a highly efficient fashion. I think that if we could design something like that de novo, which we're not at that point yet, but we might be in the future, that would be sort of the holy grail of what gene therapy could be.
And why should our audience care? I'm sure there is a lot of people that are in our audience that do care naturally, but what does the future look like as these technologies converge? What are we going to use these tools for? Why is delivery important?
Right. So there are a lot of reasons delivery is important, but really sort of the main thing is that right now our medicines are really almost all of our medicines are small molecule pharmaceuticals, and they're imprecise. They have a lot of side effects, and they don't really program our biology in a way that is very predictable or even very on target in a lot of ways. So if you take a pharmaceutical, it'll bind to not only the target receptor that's supposed to turn on a signaling cascade that affects your cells in a certain way, but it'll also bind to a lot of other things in your body and turn on other signaling cascades having side effects. Furthermore, even on the target receptor, that's a very limited type of effect that you can have on biology. So if you're just manipulating cell surface receptors and the pathways that come from them, you don't really have the ability to do a whole lot with that. And so medicine as a field, I really do think that medicine as a field is kind of in some sense in a medieval state with regards to how things are treated. And gene therapy is kind of like the next level up. Gene therapy is a way that you can reprogram biology using the source code, using the DNA and the RNA to actually affect cells in complex ways, affect them in much more potent ways, and change things. And so this can be used, for anything from Alzheimer's disease to cancer to, right now it's used for a lot of metabolic diseases. And you could also use it for treating aging. A lot of people right now are very excited about gene therapy for aging, but there's sort of this feeling that delivery is kind of the big obstacle or one of the big obstacles. Mm-hmm. And the idea of being able to reprogram the body at the DNA level, I think is very promising for being able to reverse a lot of the effects of aging. Not necessarily all of them, but I think a lot of them. And so, if you care about like a certain disease, like in your family, maybe you'd have somebody who's dealt with Alzheimer's or a mental illness like schizophrenia. If you have somebody who's dealt with cancer, or even if you have some people in your life who are getting older, I think that gene therapy really is something that has a lot of promise. And I think that gene therapy, as it matures and becomes a more widespread type of medicine, I think that we'll see a lot of changes in how it affects people and how we really might start getting dramatically better outcomes for a lot of these illnesses.
Mm-hmm. I mean, we can manipulate our source code. We can turn ourselves into a butterfly. We can make ourselves immortal. I think there's a lot of things we can do, but it's expensive, right?
Yes,
it's like these gene therapies are incredibly expensive right now. Obviously, that's like a scale issue primarily, but also they're like intrinsically more difficult to manufacture than a small molecule, which is just like organic chemistry synthesis, which we're really, really good at, right? We can, get a pack of medicines for like $3, and that's with markups where everyone's made profit along the way, right? How do we get first sort of AAVs and also, I mean, the protein vault technology manufactured in such a way and at such a scale that they become cheap enough that everyone can access them, and we can enable this transhumanist future for everyone in the world?
So I think that AAVs may or may not be sort of the end-all be-all for this, because gene therapy, as we've discussed, has a lot of diverse vectors, and I think that there is a lot of promise for each of them in different contexts. So it might not necessarily be that everyone in the world gets an AAV therapy. It might be that some people, depending on what they're dealing with, get adenovirus, some people get AAV, some people get virus-like particles. But I think in the more central theme that you're getting at is, of course, how do we produce these things? And how do we produce them cheaply enough that people can access them? And so I think that across all of the gene therapy delivery mechanisms, there's different sort of actions that can be taken, but focusing in on AAVs specifically, we need to have a way of I think that we need to leverage synthetic biology more heavily to enhance AAV production, because right now a lot of the companies that are working on AAV manufacturing are working on sort of very incremental changes in process development, in purification, in tweaking the amounts of CO2 in their cell culture chambers, et cetera. And while this can be useful, I don't think that it's going to enhance the production 1,000-fold, whereas synthetic biology has potential to really drastically alter how cells behave, how they make AAVs, and could potentially increase the yield of AAVs to a much more dramatic degree. One example is a technology called TESA, which is it actually uses an adenovirus genome that sort of turns itself off so it can't make adenoviruses, because AAVs and adenovirus have this evolutionary relationship where in the wild, AAVs actually use adenovirus. They're almost like parasitic on adenovirus. They in order for AAVs to be able to replicate, they need an adenovirus to be co-infecting the same cell, which is interesting. And so when adenovirus is present, AAVs actually infect cells much better. And that's actually another project I'm working on involving sticking AAVs onto adenovirus and enhancing their uptake into cells. But that's another story. But this technology called TESA, it actually uses adenovirus genomes that don't contaminate the resulting product with adenovirus itself, because that's a problem. If you're making AAVs using adenovirus as a way to sort of enhance the production, it can lead to contamination with adenovirus, and that kind of counters the benefit in a way. But what this group did, and now they have a company, I believe it's called Oxgene, which uses TESA. It's an adenovirus genome that you have it in the adenovirus, you infect your cells with it, and then you infect your cells with another adenovirus that carries the AAV genome within its genome. And this TESA thing, it actually turns off the production of the adenovirus capsid proteins. So it can't make adenoviruses, but it can provide the genes that are necessary to help AAV be made. Mm-hmm. And so it takes advantage of the a lot of these genes will go into the cells at the same time, and then you can reinfect these cells with AAVs, and you'll keep making them, kind of the way that you can keep making adenoviruses, which is really cool because people have been able to achieve that, 30 million patient coronavirus vaccine using adenovirus technology that sort of functions in a similar way. And this group was able to leverage synthetic biology. Even in their initial paper, they said that they were able to get up to 30-fold higher AAV production than traditional methods. Mm-hmm. And this is just one example. Mm-hmm. And so I think that this type of thing, this type of synthetic biology method has a lot of potential for AAV production, for gene therapy in general. Mm-hmm. And there's also something called doggy bone DNA, which allows you to produce more plasmids more easily, and that's another synthetic biology mechanism. So there's really, I think it's really an underexplored area, and I think that there's a lot of potential for synthetic biology in manufacturing.
Fascinating. So you're essentially providing the AAVs with the sufficient amount of proteins from the adenovirus that they can use in their parasitic ways to help them grow, but not sufficient numbers of proteins. You eliminate the sort of necessary infectious proteins that would allow the adenovirus to propagate, right? So that doesn't contaminate your end sample where you just want the AAVs.
Exactly.
It's fascinating. We are running out of time, but I have sort of one final question for you, which is, what is a book that you'd recommend to our audience, and why?
Interesting. So I've always been somebody who really appreciates the philosophical aspects of science and technology. And I've always been, like I said, a very creative kind of person. I love thinking about the big picture, imagining things. And so I would recommend a book called Spin by Robert Charles Wilson. It's actually a science fiction novel, which is sort of about the big and the small of humanity in the face of enormous challenges. And essentially the premise is that there's these kids who grow up in a world where when they're very young, all of a sudden the stars go out. And it turns out that the Earth has been sort of enveloped in this sort of shield that prevents things from going in and out very easily, or rather it sort of changes what happens on the inside and the outside. And the Earth is kind of trapped within this sphere. And it's done so by presumably these alien-like creatures called the hypotheticals. And what happens is that we see a huge global progression of different responses to this from different people. And we see it both on the very large scale and the very small scale. The individual emotional reactions, the large-scale societal changes, and how different people respond to it. And I think that this book is really amazing in that it's able to show kind of the spirit of humanity for good and for bad. And it kind of reminds us of who we are and how we respond to crises. And I think that it touches on both sort of the spiritual aspect of it and the scientific aspect of it, and brings everything together in a very beautiful fashion. And so I would highly recommend this for anyone who wants to change the world, who wants to think about the big picture, as well as who wants to think about the individuals and how things affect those specific individuals on a very deep emotional level.
Great recommendation. These book recommendations are always really good. I wish I could read 2 books a week, and then I'd be able to read all the books of our guests. Logan, it's been incredible having you on. It's been an honor interviewing you. Obviously, we didn't touch on everything. You also have worked on connectomics. You've spoken about existential hope. You're interested in mitigating AI risks, etc. So I'm sure there's room for a future episode when the company IPOs potentially. But yeah, thank you for coming on. It's been a pleasure to have you.
Thank you very much. It's been a pleasure.